fear. We all know that feeling, right? That heart-pounding, stop you in your tracks moment where your whole body just freezes. But what if I told you that freezing is just the tip of the iceberg? Today, we're going to deconstruct fear and see what's really going on underneath the surface. So let's start with a really fundamental question. How does one single scary event sometimes make the entire world feel threatening? This is at the very core of things like generalized anxiety and PTSD, where fear just bleeds out from one specific trigger and starts to color everything. What is happening in our brains to make that happen? To get to the bottom of this, we're going to dive into a really fascinating study from 2017 by Kassan and McGann. Their experiment gives us this incredible, almost startling window into how the brain learns to overgeneralize fear, how one little threat can suddenly flip a switch that makes everything seem dangerous. Okay, so here's the basic setup. It happened over about a month. First, on day 1, the researchers just let the mice get used to their environment. Totally normal. Then came the important part, the training. After that, they tested the mice's reactions a few days later, and then again a whole month later, just to see how much of this lesson really stuck. The training itself was pretty straightforward, classic fear conditioning. The scientists exposed the mice to a very specific smell, methyl valerate, and right after, gave them a mild but, unpleasant foot shock. They did this 10 times. The goal is simple, right? Teach the mouse to associate that one specific smell with danger. So here's the million-dollar question. After all that training, did the mice learn to fear only that one particular scent, or did something bigger happen? This is the question that really gets to the heart of how fear can generalize and spread. And the results were just, wow. The trained mice didn't just freeze when they smelled that one specific scent. They froze in response to any odor, even brand new ones they had never smelled before. And this wasn't a temporary thing either. This effect lasted for over a month. One bad experience with one smell had turned their entire world of scents into a threat. And this is where it gets really interesting, because this wasn't just a behavioral quirk. The change was literally in the hardware of the brain. The olfactory bulb, which is the very first stop for processing smells, had been rewired. It had become hypersensitive, basically treating all incoming smells as a potential red alert. The fear had actually changed their perception of reality. So it seems pretty simple, doesn't it? The mice learned to be afraid, and they showed that fear by freezing. But this is where our story takes a huge pivot, because this experiment and thousands of others just like it forces us to ask a much, much deeper question. What is fear, really? Yeah, we saw the mice freeze. Case closed, right? Well, not so fast. According to researcher Michael McDonald, in a big review from 2023, assuming that freezing is fear is a massive oversimplification. It's like finding one single puzzle piece and claiming you understand the whole picture. There's way more to the story. And this quote from him just hits the nail on the head. He says, "We could understand the complete neural basis of freezing, yet know little about the neural basis of fear." What he's saying is, by focusing so much on freezing, neuroscience might just be mapping the circuit for one behavior, one symptom, while completely missing the bigger, more complex state of fear itself. So to really get a grip on this, McDonald suggests we need to deconstruct the very tool scientists use to study fear. He breaks it down into 3 separate, really important concepts. But first, a super quick refresher. Pavlovian fear conditioning is the gold standard in the lab. You take something neutral, like a beep or a smell, and you pair it with something unpleasant, like a little shock. You do that enough, and pretty soon the beep or the smell all by itself will cause a fear response. All right, here's McDonald's first concept, the procedure. This is just what the scientist does. It's the rules of the game they set up. How strong is the shock? What's the cue? How big is the box? This is the part the scientist is in complete control of. Second, you have the process. Now, this is the magic, invisible part that happens inside the animal's head. It's that internal aha moment where the brain forms an association and learns, okay, this smell now means danger. We can't actually see this happen. We just have to infer it. And third, we have the behavior. This is the part we can actually see and measure. It's what the animal does when it hears that beep or smells that scent again. And this is where things get really, really interesting. So why does splitting these 3 things up matter so much?
Well, because for decades, neuroscience has kind of acted like there's only one behavior that really counts. But that's just not true. The reality is way more diverse. When an animal gets scared, it doesn't always just freeze. I mean, look at this list. Depending on the situation, it might run or dart away. It might stop trying to get a treat it was working for. It might stand on its hind legs and scan the room. And of course, its heart rate and blood pressure are going to change. Fear isn't one response. It's a whole catalog of potential behaviors. And yet, if you look at the research, the focus has been incredibly narrow. McDonald's analysis is just staggering. Over the last decade, he found 630 scientific papers focused on freezing. And in that same time, all 12 other fear behaviors combined got less than half that attention, with only 298 papers. We've been looking at this thing with one eye closed.
So this really lays out the two different ways of looking at it. The old, oversimplified view is totally linear. One procedure causes one internal process which leads to one behavior, freezing. But the more nuanced and frankly more accurate view is that different procedures can kick off different internal processes, which then lead to a whole spectrum of different behaviors.
Okay, so let's bring this all back to our mice from the beginning. The procedure the scientists chose, a small box, a pretty intense shock, that setup heavily biased the behavior toward freezing. It makes sense, right? In a tiny space with nowhere to run, freezing is a good defensive strategy. But what if they'd done the experiment in a long runway? The mice probably would have run instead. The setup shaped the outcome we saw.
So why does any of this matter outside of a science lab? Why is this distinction so critical? Because it has absolutely massive implications for how we understand and, more importantly, how we treat human anxiety disorders. I mean, think about it. When a person has an anxiety disorder, they don't just freeze. They have panic attacks where their heart is racing. They actively avoid places and situations that trigger them. They're hypervigilant, always scanning for danger. They feel this profound psychological distress. Human anxiety is this complex mix of so many different things.
And that is the big takeaway here. If we only study the neuroscience of freezing in a mouse, we might create a drug that's amazing at stopping freezing. But that drug might do absolutely nothing for the avoidance or the panic or the hypervigilance because those behaviors could depend on completely different brain circuits. To really help people, we have to understand the whole machine, not just one little gear.
Which leaves us with this final, massive question. If fear isn't just one thing, but this collection of different processes and behaviors, then just how many different brain circuits do we need to map out to truly understand and treat the incredible spectrum of human anxiety? Answering that is the next great frontier in neuroscience.